start recording. And how I'm going to organize it, I did look through, and I think the order, I think it's alphabetical, would go Mikhail, Elizabeth, Julian, and then Amanda for the cadence. So with that, I will pass it over to Mikhail to start us off. And you know what? I'm going to try and keep us honest. I will also run myself a little stopwatch to make sure we're kind of keep to the 5 minutes. All right.
Okay. Okay, can you see my screen?
Sure can.
Maybe I can hide these guys. Awesome. Yeah, so I'm Mikhail. I worked with Dr. Pierce Badera, who is a physician out of the St. Paul Heart Hospital. Dr. Egan, who is also a Medtronic scientist, and Paul Azio, my advisor on this project. And we attempted to visualize lead extractions from fixed human hearts. So what is, oh no, it won't go forward. There we go. What are lead extractions? So pacemakers, which are a very safe and successful therapy, consist of this can, which is the battery and other electrical components, and then also leads, which are the wires that actually go into the upper vasculature and into the heart. And lead extractions are the removal of these wires. And lead extractions are, not very common. It's estimated that at most 5% of implanted leads are extracted at some point over their lifetime. And it is generally a pretty safe procedure. They would be removed for reasons such as infection, if the lead is malfunctioning, becomes dislodged, things along those lines. And again, it's generally a pretty safe procedure, but in some cases, especially when the lead has been implanted for a long period of time, you'll get a lot of this tissue growth over the lead body, which is kind of what you're seeing here in the second image, and what they're attempting to show in this animation. And so because of that, we have to use fancy tools to help us remove these leads from the body, which again is essentially removing or separating, I guess, the tissue that has grown over the lead body from the vasculature and the heart tissue. So imaging, and that's kind of really all types of imaging, but today I will discuss computed tomography or CT and intracardiac echo, which is a type of ultrasound, makes lead extraction more safe. And that's been really heavily studied in the literature. As I mentioned, lead extractions are relatively safe, but the complications have a high mortality. And so the imaging can assist the extractor by showing what is happening at that lead tissue interface. And so we're able to get some perspective into how the lead body, the device that we are attempting to remove, is attached or connected to, again, the vascular and heart tissue. And so typically a patient will undergo a pre-extraction CT scan. That's for looking at that lead tissue interface. And then during the procedure, they can use this intracardiac echo, which is where they would insert this probe into typically like the jugular vein, which is a neck vein, and kind of move this probe into the heart to get a really kind of close up view of the lead tissue interface. And so I really just want to show this video, which is what we recently published in Heart Rhythm. Essentially, we connected some of these fixed human hearts. Here, I want to pause it so I can talk through this. Fixed human hearts up to a pulsatile pump We stuck in cameras so that we were able to see the lead tissue interface directly. This is our intracardiac echo, so this is an ultrasound imaging modality. The circle in the middle is the probe. This bright spot right here is the lead body, and this kind of goofy looking blob is actually this leaflet right here. So it's kind of just demonstrating that intracardiac echo can really give you these close-up visualizations of the lead tissue interface. And what's really interesting is before we hooked the hearts up here, we did a pre-procedural CT scan, which is this image right here. And you can see the lead body right in this corner of the right ventricle, and essentially that same image as the ice with this leaflet right here. So the point of this kind of paper that we talked through is, you know, we can learn by comparing the direct visualization to the clinical imaging, you know, what's actually going on inside a patient's body. And also we wanted to correlate the intracardiac ultrasound, which is feedback that you're getting during the procedure, to the pre-procedural imaging to determine, you know, maybe you should have known about this before you went into the operating room, or giving you like more information about the lead tissue interface intraprocedurally. So I think I'm probably at time, but I'll just play the rest of the video while there are, if there are any questions.
Yeah, Mikhail, can you, what about the tissue types? Like are you able with the pre-imaging to know what kind of adhesions or, you know, what kind of tissues you're going to be up against as you extract the lead?
Yeah, no, that's a great question. So there can be, again, with leads that have been implanted for a really long period of time, there can be these calcifications, which is the really, really dense tissue that forms right over the lead body. And you are able to see that in the pre-procedural CT because it's so much more dense. It doesn't attenuate the CT scan rays as much, and so you can see that beforehand. On the ultrasound, I think it's a pretty comparable to the vascular tissue. I think that the purpose of the ultrasound is really kind of a more, you know, during the procedure, am I in a spot where I can see the lead body completely floating in? So here's, for example, our SVC, so the vessel that leads right before the right atrium. If the lead was kind of more in this area, we would know that it's completely floating in the vessel and therefore not attached to any tissue. And so we could proceed with that part of the removal much more quickly. We would know that we wouldn't need to be as cautious in that area.
Yeah, any questions from the rest of the panelists? Do you know how often, before you mentioned a statistic of about 5% maybe of these leads are removed? Do you have any numbers on how often pre-procedural imaging is done ahead of those extractions? And the reason I guess I ask, I get feedback many times about pre-procedural imaging, and physicians say, you know, I would only do it if I knew it would change my approach. So do we know how often people are doing it?
Yeah, good question. I would say it's definitely not routine in most of the centers, and it's very unlikely in a lower volume center. So maybe not like an academic hospital, maybe more of like a community hospital setting. They would very unlikely do pre-procedural imaging, probably because they just don't have like access to that equipment, or it's, again, maybe not necessarily worth it to see what they're up against. But that being said, those that are a lead that has been implanted for 20 years is, I can tell you that it's going to be difficult to remove. And so I think before imaging or the procedure is even really planned or considered, they would likely be referred out to a higher volume center. And I would guess pre-procedural imaging is done maybe 50% of the time in higher volume centers. In some centers, it will be every patient no matter what. In some centers, it will be, like you said, only when they think it will provide some extra additional information. But yeah, we're trying to, there's definitely a case being built in the extraction literature that says this is something that, should be done before every procedure because it has a positive impact on the outcomes clinically.
All right, thank you.
Awesome. Thanks.
Yeah, so Elizabeth, you could tee up your presentation.
You can see it all right?
Yes, looks great.
Okay, so I'm Elizabeth. Again, I'm a fourth year. And today I'll be presenting on a perfusion bioreactor for improving cardiac modeling. So the motivation behind this is that we know cardiac disease is a big issue globally, and in the United States alone causes over 650,000 deaths each year. So to better understand these diseases as well as potential therapeutics, our lab has developed the human chambered muscle pump. So this structure is taken from an MRI scan of a human heart, scaled down to about mouse size, and then altered to create this U-shaped chamber. The structure is then 3D bioprinted using human-induced pluripotent stem cells, or iPS cells. And these cells are embedded in an extracellular matrix-based bioink. And then the cells proliferate for 2 weeks and then are differentiated within the structure to cardiomyocytes. And so this process of printing and then expanding to get larger numbers of cells and then differentiating in place to get cardiomyocytes allows us to obtain a high amount of various structural proteins, including different receptors that are important for signaling. And those ultimately allow for pretty high functionality within this structure. So you can see here with optical mapping, we can see signal propagation throughout the entire structure. And that leads to macro scale beating, which actually allows the structure to pump fluid from within. And so this model is pretty promising for better understanding different diseases. It's got a relatively high degree of maturation. But when we look at a cross-section of the H-champ, shown here where the nuclei are stained blue with DAPI and the cardiomyocytes in red with troponin T, you can see that there's very few cells on the interior of this structure, despite the fact that we printed with the iPS cells throughout the entire structure. And so this is likely due to diffusion limits where nutrients aren't able to access past that first 100 to 200 microns of the tissue layer. But previous studies have shown that these diffusion limits can be increased with fluid flow up to 500 microns. And so the goal of this project is to perfuse not only the interior, but also the exterior, or allow fluid flow around the exterior to obtain cardiomyocytes throughout the full millimeter wall thicknesses. And so the goal would be that this improved cell viability will enhance their functionality even further and kind of push the limits of how thick of an engineered tissue we can get. And so to help us with that perfusion, we've developed a bioreactor in collaboration with the Center for Engineering Complex Tissues out of the University of Maryland. So with several iterations, this is our current design, where the H-champ sits in a little chamber in the middle of the bioreactor. A pump moves media into and out of the structure, And then we actually have a tubing to connect the HChamp inlet to the bioreactor to kind of help force that fluid in. It's a pretty delicate tissue since it is extracellular matrix based. And then there's an external perfusion channel to allow media to move around the outside of the HChamp. There's these support posts on the bottom half that help to align the HChamp with the bioreactor openings and to allow that fluid to again move around the exterior of the surface as well. And so after 5 days of perfusion, this is another cross section, this time DAPI shown in white, just for better contrast. You can see that there's a lot of colonies around the exterior of the surface and a few within the middle. Compared to a HChamp cultured under static media, the colonies are really only residing on the outside and only part of the outside really of the structure. And if we zoom in on the, I guess, septal region between the inlet and outlet channels, you can kind of see it better here. Not only do we have 2 times larger colonies in this perfused structure, but also 6 times the number of large colonies just in this internal region. And so again, this is after only 5 days of perfusion. So you can imagine by the end of our 2-week proliferation phase that the difference would be a lot more significant, hopefully. It's a slow process, so we're hoping to get there soon. But yeah, you can imagine that tissue would be quite a bit thicker, and again, will enhance our functionality even further and allow us to use this model for, again, better studying diseases or again, potential therapeutics. So yeah, with that, I want to thank my lab members and again, the Center for Engineering Complex Tissues in helping with bioreactor design. And I'll take any questions.
All right, very cool. I wonder, so you think about the cardiac, like our perfusion and some of our, you know, you think of our coronary arteries and how they penetrate into the depth of our cardiac muscle. Do you think that that's going to end up being one of your refinements, that there needs to be something that goes intra, like through the thickness of your printed geometry?
Yeah, for sure. And there's another member in our lab working on kind of optimizing endothelial differentiation and looking at how we can incorporate that with the cardiomyocytes.
Okay.
So I guess the other aspect, once we do get to the endothelial stage, later on, post-differentiation of these cells, we'll kind of be adding a fluid biomimetic stimulus that will help to mature those cardiomyocytes. So that's kind of a whole nother aspect that we'll look at too, that will be beneficial post the viability stage.
And is there a point, your first couple slides you talked about then kind of the symptom, they do start to beat in concert with the cells. So when does that, like for instance in these slides that you're showing us here, does that start to happen yet? Like so at day 24, so when do you start to appreciate any sort of mechanical function from or electrical, like synctium beating, if you will?
Yeah, and these structures, I'd say beating starts around day 10 or 14, somewhere in that range. So yeah, right now, the results I'm showing are still clear back here in the proliferation phase with the stem cells. So yeah.
Can I ask a question on how the optical mapping works? I think I knew that at like some point in my life, but I don't remember now.
Yeah, so I mean this, oops, it's gonna freeze on me, but that video was taken by the people that have worked on this initial development stage. I haven't gotten to that part in my research yet, so.
Okay. Is this the thing where the heart kind of hangs and you spin the camera around it? Is that optical mapping? No.
I
Okay.
really
I don't
know.
know. That's okay. I mean, you could tell me anything. So okay.
Some of it is I mean, the optical mapping that's used in electrophysiologic studies is a voltage sensitive dye. I know. So there's
Okay.
probably a few different versions of optical mapping, but one is sensitive to the transmembrane voltage, the cells. So it's you know, the optical mapping is usually done before there's mechanical motion to mess up the image, and it just has a voltage proportional to the cell, the depolarization of the cells. So I know that the work that comes out of St. Louis and Cleveland Clinic was based on that form of optical mapping.
Yeah, I think that's what method we're using too. Yep.
Okay. All right. Thank you, Elizabeth.
Yeah.
All right, we're moving on to Julian. I'm just like watching the time so we don't shortchange Amanda at the end. So thanks for the presentation, and I'll turn it over to Julian.
Is good? Okay.
Yes, it is.
So today I'll be presenting some research I've been working on recently titled Synthetic Encapsulation Technology to Model ER Dynamo-Regulation During Immobilized Induced Dormancy. So some background, first of all, on biomarker discordance. So this is when the primary tumor and the metastatic tumor have different biomarker expression. So it can either be that the metastatic tumor has an increased or decreased amount of a certain biomarker. And this difference can help explain why there's a wide variety in the response to treatments in patients. Typically, only the primary tumor is examined, and the metastatic tumor, which later develops, is treated similarly to the primary. And if there's a difference between those biomarkers, you might be treating for something that is not going to be effective. More specifically, I'm focusing on ER, estrogen receptor, which is a common target in breast cancer. And typically, estrogen receptor positive cancers are easier to treat. There's a lot more treatments because there's molecules that will go and target that receptor and can either slow down or cause the cell to apoptose. And then the discordance rate in the estrogen receptor has been found to be 21%. So this is either the primary tumor was positive and the metastatic tumor was negative, or vice versa. And then this all relates to dormancy, which is occurs when some cancer cells basically survive in metastatic state dormant for months or years, and allowing them to stay in this state helps them evade a lot of the therapies. And this dormancy typically occurs in breast cancer patients who have ER-positive tumors. So the primary tumor is taken care of either through surgery, chemotherapy, or radiotherapy, but some cells are left behind, typically in metastatic regions, and those cells can later awaken and become tumorigenic. And so we have been working on developed an immobilization technology to basically keep cells in a physically immobilized state where they cannot move or proliferate. So to the right here, the cells are first coated in a thin layer of agarose using a microfluidic flow focusing device. Then they're collected and encapsulated in a silica gel that keeps them completely immobilized, so they cannot move. So this produces a stress that can isolate the cells that can only go dormant. So this leads to a very dormancy-like response as what's seen clinically. So we were interested in if undergoing this dormancy process can replicate some of what's seen clinically in terms of this biomarker discordance. And we focus on ER given its prevalence with dormancy. So I first looked at immunostaining of cells against ER. So our control group was cells grown on tissue culture polystyrene, and these cells were predominantly ER positive, and as we would expect. And then cells that were immobilized in our MSPEC, which is a silica gel, for 3 days. And what we saw is that there's a significant reduction in the number of cells that are ER positive. So I confirmed this with Western blot analysis. Here is ER expression and also beta-actin. And again, we use tissue culture polystyrene cells as a control. So there's this, this is mislabeled the other way, sorry. But we still saw a 65% decrease in expression after 72 hours in immobilized. And finally, to confirm that the remaining ER was not functional, we treated cells in the gel with estradiol, which is the hormone that will binds to ER and causes proliferation. So if a cell tries to proliferate while immobilized, we have observed that it leads to the cell death. So we measured viability in the gel at various concentrations with estradiol, and we saw that none of the concentrations produced any effects. So from this, we concluded that the ER wasn't activated. Otherwise, it would have led to proliferation, which in turn would have led to cell death while immobilized. So in conclusion, ER is an important biomarker in breast cancer, and we showed that cells, MCF7 cells, immobilized in our silica platform are downregulate ER, and the future work will focus on other biomarkers and also determining the mechanisms of this ER downregulation.
All right, thank you. So, a question on, how this might change, current treatments. Then is your work really, the mechanism question of what would happen, or is there treatments that are really focusing right now on this immobilization induced dormancy?
The way I see this platform is, so typically it can be months or years before a patient, the dormant cells awaken and become tumorigenic. So if you could identify from the primary tumor when it's removed, identify if there's any dormant cells, and you can isolate them and figure out, basically characterize them to figure out if this patient is more likely to have ER positive or ER negative dormant cells awaken later, or figure out if there's a better treatment. So leading more towards the personalized medicine.
Okay.
Yeah.
Thinking about just, been talking about the imaging and, is there work that you've done, or the imaging work and some of the staining work that you've done that there's been a revolution in how that imaging is being done in your lab or in the clinic? What are the big changes?
With our gel, it's a little bit hard, and I tried to pick obviously the best looking pictures, but some of the images, because this gel is opaque, so it is a bit difficult to image, but with fluorescence, you can get good enough images. These are very thin layers. These are 100 micrometers approximately. so that helps. But for a good chunk of my PhD, I had to basically plate the cells in a kind of like a sandwich method to be able to visualize, because it's very difficult to get. You'd have to go into confocal and more fancier techniques.
Okay. Yeah, it's just, you know, I always appreciate how much time and effort go into, you know, making this panel of data that we see here, right? There's a lot of plating work and imaging work to show us these final results. And just wondering if you've seen differences in that technology over time and how it makes, you know, conducting this research less tedious, less time under the microscope, less time
I mean,
under
I
these
think in
fluoroscopic
terms
techniques.
of the imaging hasn't been too as bad. The quantification, just because there's so many cells. There's programs that work very well with this that can, it's mostly identifying like this is a cell and then layering on top basically if this is a cell, is this also positive and negative? So using a threshold just to automate it, because doing this manually is the most cumbersome.
Yeah. Okay. Any other questions, Julian? All right.
Actually, I have a very quick question. Sorry, I was wondering if you could speak to on one of the first slides when you were showing your process for that slide for preparing the cells. Could you speak to the role of the magnet in there?
Oh yes. So another huge challenge in this whole, the original gel was just silica and PEG, polyethylene glycol, and it was really hard to get the cells out. So I worked around that to basically still keep as much of the original system as possible to keep them immobilized, but be able to recover them. So basically put them in agarose that protects them, and then thorium iron oxide to get rid of the silica. So this gray particles are the silica. So the idea is the magnet just to be able to get rid of that.
Sure, cool. Thank you.
Great, thanks, Julian. And then Amanda.
All right. Cool. Okay, can everybody see the slides?
Yes.
Awesome. All right, so hi again. My name is Amanda Tenhoff, and I will be presenting today on patient-specific 3D printing for interventional planning and patient education in complex branch pulmonary artery stenosis. So the case I'll discuss today was complex branch pulmonary artery stenosis in an 18-year-old female patient. She was diagnosed with truncus arteriosus type 1 earlier in life and had received several surgical interventions since infancy. However, development of recurrent conduit stenosis and residual pulmonary arterial stenosis occurred. Important anatomical considerations when making the 3D model for the preceding intervention are shown here. So starting in the axial view on the right, there is significant occlusion of the left pulmonary artery ostium as a result of restenosis. Additionally, through discussions with the lead clinician, it was determined that the stent in this area contained extravascular portions, which were not initially apparent from the CT scans alone. Next, the coronal view indicated significant stenosis in the main pulmonary artery parent branch, as well as the PA conduit shown in the sagittal view. This information was then used to create the patient-specific 3D model of the conduit and main PA branches. The patient's CT scans were imported into materialized Mimic software for segmentation of the blood volume from the tissue in the area of interest. Next, this model was smoothed shelled, and further refined in materialized 3-matic software. So models from 3-matic are shown here, with the left image showing a smoothed and shelled opaque model, and the right image showing a smoothed and shelled transparent model to visualize the embedded stents. From here, a hollowed model was printed on an Ultimaker 3 printer at 100% scale using PLA, and this was then delivered to the clinician and the care team. Due to the unique internal features of this case, collaboration was sought with Stratasys for printing a flexible plastic version of the model with stents, visible stents inside. Additionally, another model was printed that was the same size, 100% scale, and hollow, flexible plastic. The purpose of all of these models were to gauge proper balloon and stent sizing, as well as catheter approach technique. Additionally, this model was especially important for increased patient education and understanding regarding her disease state and treatment options. So it should also be noted that these initial prints did not contain the correction pertaining to the extravascular stent. However, these models still prove to be useful for understanding the complex anatomy of the case and interventional planning, as well as discussing the diagnosis and treatment options with the patient herself. Based on these models, the clinical decision moving forward was double barrel stent deployment in the distal conduit and proximal branch arteries. Shown in the figure on the left, depicting a contrast CT angiogram of the PA conduit and left and right pulmonary arteries, blood flow was greatly occluded and the resultant right ventricular pressure was 65% of the systemic blood pressure. After deployment of the stents, the proximal stenosis was greatly corrected and beneficial effects on the blood pressure were seen immediately, decreasing the right ventricular pressure from 65% of the systemic pressures down to 35%. In addition to the evident procedural efficacy demonstrated by these clinical outcomes, the patient was quoted later describing her increased quality of life. She said, "I have never felt this good in my life. I am enjoying it." And I think I can speak for all of us working on advancing medical technology when I say that truly, at the end of the day, this is why we love what we do. So to wrap up, creating and utilizing these patient-specific 3D anatomical models is immensely useful in understanding patient anatomy, especially in patients with complex congenital heart disease. Utilizing these 3D models for simulating the intended intervention techniques helps to formulate an intervention strategy and lead to measurably beneficial patient outcomes. Additionally, these models are particularly useful for aiding in meaningful interaction and discussions with the patient and families to explain the given pathology. Going forward, patient-specific treatment strategies will be assessed through computer simulation and virtual interventions, in addition to these 3D printed models. We believe this approach will be the future of medicine. And with that, thanks for your time, and I welcome any questions.
Thanks, Amanda. Question on the model itself. So you showed that the outcomes from it lowered the right-sided pressures by a certain percentage. Does your model, does the modeling effort try to calculate flow or pressure differences? I guess did you have a target amount that right-sided pressures would lower, or?
Sure. I'm really glad that you brought that up. That's actually kind of the future direction for myself and my research. But what I'm presenting today, the answer to that question is no. This was purely an anatomical model. Okay. So it was just for viewing the anatomy and making inferences off that. But yeah, I'm glad that you said something about that, because in the future, simulating those flow patterns and understanding how different interventions affect resultant pressures is going to be my the driving idea for my research going forward.
Okay.
So I have a question, Amanda. Nice to see you again. So did the cardiologist change his or her approach to this particular case based on your model? I didn't quite get that. I know it was used with the patient discussion, but did they actually like try their procedure on this model?
That's a great question. I don't know for sure, yes or no. I know that that was the intended purpose. So that I worked on this case with Dr. Varun Agarwal, and I think that he did use the model to assess stent sizing in actual stent, but I don't think that he used the models to deploy the stent or like practice deployment. He may have, I'm just not aware if that was the approach he took.
Thank you.
Along those lines, I guess kind of, did you have a way to assess how close your print like sizing was to the original? Like, did it reflect pretty well upon the original?
Yeah, thanks for that question. So whenever I finish creating the models in 3matic, because this is kind of the final step before they would then go to printing, I import it back into Materialize and then superimpose it on the CT scans to make sure that everything matches up okay.
I have one more question. So can Stratasys actually print a material that emulates a stent versus normal arterial tissue, or is it just kind of cosmetic that you can see that there's the pattern of a stent, or is it really simulating a nitinol?
That's a great question. I can't speak exactly to whether or not they're able to simulate properties of metal, but this model, yeah, this is just a firmer plastic than the rest of the actual anatomy around it. So it's a harder plastic inside a softer plastic, and then that was the stent model then.
Thank you.
Mm-hmm.
Okay. Well, thank you, Amanda.
Yeah.
I think, well, Dr. Audie's here. I think the next section of this is to just look at ways that that very different, like 4 different very sort of presentations. What are some of the possibilities of collaboration and the things that we found that speak to all of these projects? Like in a similar way, I certainly think that patient-specific medicine, like all of these cases, sort of speak to how we are personalizing, whether it's for congenital patients or patients that are having complications from lead or device implants, or the ability to grow organs or replacement tissues or grafts, to some of the work that on diagnosing, and isolating problems like from tumors or cells gone awry. So, I think personalized medicine is one of those things that is coming slowly, and we see little kernels of examples of, but it will, 10 years from now, I'll probably look back and see all these things kind of converging and coming together. I don't know if for this section there's certainly some questions about the opportunity for the pilot project as well, and I'm going to bring that slide up. I will hand it over to the other moderators, see if they want to direct this next section of discussion. Let's see.
Well, I'll do my best since I missed the first 3 or 4 talks. Sorry I was late, everyone. Schedule conflict, but I'll give it a whirl. Oh, okay. Money involved. There we go. So, right. I think this was kind of piloted in a slightly different format last year, where one of the goals of the annual conference was to try and synergize and create some new research opportunities. So in fact, a person in my group, a postdoc, was a winner last year, and I don't know exactly what she did, but she was working with somebody else I never heard of on some completely different project. So that's
Yeah,
great.
cool.
That's the beauty of the university, right?
Right.
So, right, I guess the idea is to have at least, you know, you and one other person maybe on this panel get together and try and synergize with a new idea. So I guess maybe it would be good at this point to kind of open it up to the presenters, like, okay, Amanda, for example, was anything presented by someone else that you thought, oh, you know, here's a connection I wasn't aware of. So not to pick you out, Amanda, but since I heard your talk.
I probably have to think about it for a little longer. But I know that we have to be in, well, that's a good question. Do we have to be in different labs, like research labs?
I don't think so. Certainly, this is sort of the level of the rails I think that I was given on this. That I think, you know, it again it's probably things like, you know, listening to the 4 different talks, and many of you are taking like even I would say Elizabeth's bioreactor. I mean, you're starting with an MRI or a structure, a cardiac structure, and you're growing your cells, or you're doing your work off of something that is coming from a scan. I think, and that's true for Mikhail's work, as well as what Amanda was presenting on the anatomical structure that is probably working on some of the functions. So I don't know if there's some ideas around, you know, like imaging tips and tricks and overlays and these models that you're developing on functionality of the cells, of the cardiac cells. There may be some work in there from the extraction work. I know, you know, there's a question. I can see that there's an opportunity for simulation and of what makes leads and devices react, or different people react more violently, if you will, to devices that are implanted in the heart. Some are very hard to remove after, you know, after a year, others are not. And doing that kind of very applied research and like implanting something, let's say in an animal model, and waiting a year to explant is a pretty brute force approach probably to the problem. You know, if we can grow those cells and differentiate those cells on the bench, we can probably then come up with strategies on how to best remove devices, or capitalize on the other hand. If you want something to grow in very quickly and wall off, like, you know, one of the things that I've been working on for years is little devices for epicardial pacing. You know, maybe it's something that you want, you know, very quickly to be walled off and protected. So it's, you know, you're not worried about infection or other things. So, you know, I think there's intersections between what I heard today and, you know, what might be a pilot on these different ideas. The bioreactor is very interesting to me on like, how do you, you know, how do you grow different cell types, or how do you, you know, grow a model that then you can work with, like it's the full thing, right? You can design, understand what happens to your hemodynamics, perhaps when you're growing something that looks more like a congenital heart.
Yeah, so just listening to Teri there make the connection. I mean, I agree, not to exclude Julian and is that Michaela there from the conversation, but yeah, the idea of some CFD-based project that would help both of you. So Elizabeth, I didn't hear the details of your bioreactor, but it, you know, clearly Amanda, yours is going to involve like the classic fluid structure interaction problem. So you have a deforming pulmonary artery or branch as the blood pulses through it. And so I'm not sure if your bioreactor, Elizabeth, would involve, again, a deformable material, whether it's your cardiac tissue or something else. But I mean, there might be some way you could work together on using ANSYS or something to try to model some related problem that's of interest to both of you. I do see that on the slide, right? You don't have to be working with somebody on this particular panel or in this session. You can scan the whole list of 70 abstracts and say, oh, gee, that looks like it's pretty close to my interest, and just contact that person and see if you can come up with again, a 2-page proposal, get you a little bit of supply money. Yeah, you can moonlight from your thesis project on the weekends. What a deal.
Right. Like ideally, it's something that I know for those of you, some of you who see the finish line here pretty close in your sites, right? I think many of you are finishing up your PhD, but it maybe an opportunity for that seed money for a postdoc or but I think those of you who are in your second or third year, what are some slight things that would help perhaps your PhD, whether it is tools and tricks from another lab on how they're doing their auto segmentation of their CT scan, like how do you get your anatomical model done more quickly versus Julian, I know like you're saying it would be pretty horrible to be counting cells and doing a lot of things by hand, and the automation tools and tricks have improved over the years. So my encouragement is at least to pick up from the different labs something that they're doing better than your lab. And like, I could use that, or I could morph that into something that would speed up my project or help me go down maybe a slightly different path for an application. So I will say the instructions that I got, we will, the sessions, I think you have an opportunity to see other presentations. So this right now is being recorded. So you're able to go on, and I think there's 13 or 14 different breakout sessions. I may have my numbers wrong. So you're also able to go and see people's 5-minute presentations, see what else is out there. And maybe, I know some of you are in the same labs, but there's probably representation from must be several dozen labs from across campus. So I'd encourage you to take a look to see what else is out there and reach out. Now, my understanding, everyone should have from the program a list of emails and contacts for all of the presenters, right? So I would use that. I would use the facilitators as well.
Right, I'm looking at an Excel file that has at least the names and a link to the abstract.
Okay.
But, yeah, so, right, should be like being in a candy store, right? You get to go around and shop and find something that maybe you want to invest in.
Yep, good stuff.
Yeah.
These are great opportunities, I know, to pick out collaborators and see what else, see what someone else is working on that sparks your interest as well. So, any other comments or questions? We're about it. I know I don't want to run over time, so I know we're about at the hour. I want to see if you got any closing questions or comments.
So
no, this is my first virtual poster session, so it's an experiment.
Are faculty members eligible for this $5,000?
Probably not. I don't think so. Although you might be able to, get one of your grad students to work on something that's of mutual interest to you both.
I was being facetious, of course, but I guess what I found interesting, here you go, right? So we're trying to grow we're creating conduits that grow for pediatric heart defects. And our first clinical trial is aimed at discontinuous pulmonary arteries and hemitruncus. So there you go. I didn't know Amanda was working on something that, could provide a surgical model to help us figure out exactly what size and shape, et cetera, we should aim for. So there you go.
Right.
Lesson learned.
Awesome.
And I will say that I have an interest, of course, in device extraction and better ways to do that research, that doesn't take a long time. So I am very interested in the bioreactor work, and I thought you guys all did a nice job of because we're from disparate backgrounds, and you did a nice job of walking through your research and giving us, a very fast elevator pitch of what you're doing and why you're doing it, and the clinical relevance. So I applaud that. So thank you for
Okay.
your work and putting it together.
Thanks for leading it,
Okay.
Teri. Appreciate
All
it.
right.
Thank
Okay.
you all.
Thank you
Good luck.
very
Bye.
much.
Bye.
Bye-bye.
Everyone.
Take care.